dimorphism or in these elaborate traits that we see in secondary sexual characteristics. But not all sexually dimorphic traits are this grand and this beautiful. Sexual dimorphism actually occurs on practically every level of organism physiology. And so to show some of those less pretty examples of that, we see that in mice, the immune cells of adaptation show very dimorphic levels of gene expression of pathogen recognition proteins. And so there's sexual dimorphism on the cellular level and on the expression level here in mice. And in these hoverflies, there's even dimorphic neurobiology shown here by their response of visual neuron to the same stimulus. And so sexual dimorphism is happening across many different levels. And what I'm really interested in is how these traits, these sexually dimorphic traits, evolve. And so sexual dimorphism typically evolves because males and females have different optimal values for many traits throughout the genome. However, males and females share the same genome, and so this presents a problem with constraints to the evolution of these traits. And given that males and females share the same genome, same genes, we know that these traits come around by the differential expression of the same genes. So what I call here sexually dimorphic gene expression. I'm going to use that little acronym there to keep it simple. And so I'm really interested in how sexually dimorphic gene expression evolves, and we've learned quite a bit about this over the past decade, and some of the mechanisms are very interesting. There's a couple different ones. What we know is that in all metazoans, a gene in the DMRT family, double-sex MOB3 related transcription factors, initiates male and female gonad development. And in vertebrates, the gonads secrete sex hormones, which then leads to the differential regulation of gene expression. In invertebrates, however, they don't possess these sex hormones, and so instead they utilize, probably utilize spliceable variants of the double-sex gene. And in fact, we see a lot of these binding sites throughout the genome that support that these mechanisms can differentially regulate the genome. So though these are the mechanisms that we know kind of the most about, there are probably many different mechanisms and other mechanisms, and there's one that's really obvious that still needs to be tested, and it's that the sex chromosomes are directly influencing sexually dimorphic gene expression. So specifically the heterogametic sex, Y chromosomes, the XY systems, W chromosomes, the W systems, have some direct effect on regulation of genes and lead to sexual dimorphism. And that's because these chromosomes are sheltered from sexual conflict. They only exist in one sex. However, when we look at the characteristics of those chromosomes, it doesn't seem to be intuitive, doesn't seem to be obvious. These chromosomes are very heavily heterochromatic, they have very few protein-coding genes, and there's really no regulatory function on these chromosomes. So people said, all right, it doesn't look like the sex chromosomes are leading to this differential expression of the genome. However, recently, we found out a little bit more about the Y chromosome, and that it can actually regulate the genome, not through the protein-coding genes, but through Wily regulatory variation. And Wily regulatory variation is driven by the heterochromatin, or the chromatin landscape throughout the genome. So Y chromosomes may be variable in the amount of heterochromatin they have, and those have downstream effects on heterochromatin expansion and positioning throughout the genome, changing the regulation of the entire gene. And this effect is not just Drosophila specific. In fact, we think it might be very widespread, as most Y chromosomes and W chromosomes are heavily heterochromatic. They have variation in their heterochromatin, and in an independently evolved murid Y chromosome, we see these Wily regulatory effects. So essentially what I'm saying is the Y chromosome can now provide that sex-specific variation that can lead to differential gene expression. Selection could shape that, and we could get facilitation of sexually dimorphic gene expression. This could be a mechanism by which we differentially regulate the genome across the sexes. In order for this to be true, though, selection has to be able to act on YRV. For selection to act on YRV, it needs to exist within natural populations, it needs to influence fitness-related traits, and it needs to be comprised in part at least of some additive genetic variation. None of these questions have been addressed so far looking at YRV. Typically, the research is looking at Y chromosomes in very distantly related populations and looking at gene expression for nothing of real fitness or things that we don't really know the fitness consequences of. So my research is focused on those questions. To get the first one, does YRV exist within natural populations? we went out and we collected wild fruit flies from Orlando, Florida. We got 30 different males, and we took those males and we placed their Y chromosomes into a single cloned isogenic background. So essentially these different lines all have the same genome, but different Y chromosomes. And so we can look into those lines and try to determine if Y chromosomes or YRB is existing in this population. And so we took those lines and we tested for the expression of 3 different immune genes, the qPCR. We picked these genes because previous studies had detected that different Y chromosomes from different populations influence these genes. And these are primers that we already had in the lab, so that's pretty convenient. And we looked for the presence of YRB in this population. And so what we saw were that females of these lines, which contained the same genomes, they were isogenic clones across the lines, showed no variation in their gene expression, and that's what we'd expect. The males, however, which differed only in their Y chromosome, showed variation in the expression of 2 of these 3 genes. And I'll make sure I point out these are autosomal genes, these are not Y. So Y chromosomes that have no immune genes on them and no regulatory protein-coding material have resulted in differential expression of a couple of our genes. To see if this had any kind of fitness consequences, since we're working with immune genes, we said let's test their actual immune response. And so we took 30 different Y lines, and we infected them with an LD90 concentration of Serratia marcescens, and that's a gram-negative bacteria. And then we measured the response to this bacterial challenge. What we found, and the response we were looking at was their time to death, which we were able to measure with these activity monitors, which is quite nice. Again, what we found were the females across those lines were not different. That's what we expect from clones, but the males showed variation across lines, only differing in their Y chromosome. So what these data, what was really interesting is when we took the data and we compared the time to death data to the gene expression data. We saw a really nice correlation between gene expression and your ability to survive an infection. And so you can see that lines that have very high levels of immune gene expression across these 3 genes lived the longest, and lines that had low levels of immune gene expression died earlier. So what this data suggests is that YRB does exist within this natural population, that Y chromosomes influence the survival of these organisms, and what we can say is it looks like Y chromosome is influencing this fitness-related trait. So, jumping around. So, we know that the variation exists in a context in which selection can shape it, but we don't know if it's additive. So that's what we wanted to ask next. Is it additive or is it non-additive? And this is very important because selection can only shape additive genetic variation. And in fact, previous studies suggest the YRB effect may be largely epistatic. And what we see from an earlier study in 2001 by Chippendale and Rice, looking at male fitness, that certain Y chromosomes impacted male fitness. But the effect was completely changed when it was placed into a different genetic background. So that suggests that the Y chromosome effect is largely epistatic. This trend was seen again in 2010 in a study that looked at gene expression. The effects of any given Y chromosome depend on the background that it's placed in. However, these studies had a couple caveats for kind of making the conclusions we want. The first one was in a male-limited trait, something that we may expect to increase the epistasis between Y chromosomes and autosomal backgrounds. And the gene expression data was done with elements that were non-co-evolving. They were from different populations. So we wanted to address this to kind of remove these caveats. So we took our wild lines from a single population, and that means that the Ys in the background were co-evolving. They experienced each other. And then we picked a male and female shared trait, again, a immune function, something that most males and females have to do. We took the Y chromosomes, we took 4 different ones, and we crossed them into 4 different autosomal haplotype backgrounds to look at the interaction between Y chromosomes and backgrounds. So we made every possible combination there, and we measured again the response to different bacterial challenges, gram-negative and gram-positive. And the response we were looking at was time to death. Our results showed that the Y chromosome only showed an effect in gram-negative bacteria. Gram-positive, there was no YRB for the response to that trait. But in gram-negative, which we tested earlier in previous studies, there was a Y chromosome effect. However, the additive component of this kind of matrix showed no significant variation. The Y chromosome effect, the additive Y chromosome. The Y by background effect, which is our non-additive, the epistatic component, was significant. When we estimated the genetic variance components of these different genetic elements, we estimate that the epistatic component makes up about 43% of the genetic variation for survival to gram-negative bacteria, of the genetic variation, which is a significant amount of the genetic variation. And so what this really suggests to us is that though Y or V exists in a context where selection could shape it, it's lacking the additive genetic variation required for selection to shape it. However, I got a little asterisk there because there's a couple things that we need to consider. The first is that selection works on populations, not on the few chosen lines that we picked. So even though we did four chromosomes, 4 Y-chromosomes, 4 genetic backgrounds, that doesn't include or doesn't take account for all the variation that's out there in the population. So we know that, and even more is we fixed the frequencies of these genetic elements to be equal. We know that allele frequency is very important when you're measuring genetic variance components. Epistatic genetic variation can change if allele frequencies change, it can turn into additive genetic variation. I know that's a single locus there, but let's go and check fine. But even given these aspects, if we account for these aspects, there may actually be some additive genetic variation in the population. If we took the whole population, if we accounted for realistic allele frequencies, there may be a little bit of additive genetic variation. But what we think is, given our experimental design, and seeing that there was a lot of epistasis and very little additive, that even if it exists out in nature, it's probably at low levels of additive genetic variation. So take home there from that is that additive genetic variation may exist, but at low levels is what we think. Okay. But what's more interesting is that there's a large amount of epistasis, and we know that there are some costs to epistatic genetic variation. First, it can increase the ruggedness of adaptive landscapes, leaving groups marooned on these fitness optimums that are spread out on the rugged landscape and not letting them achieve a global optimum. We also know that epistasis increases phenotypic variation. So this figure here shows essentially 4 Y-chromosomes, the different lines, across 4 different genetic backgrounds. In the top figure, everything's acting additive. And so we take that little model there, and we estimate the genetic variance components. B and C are varying amounts of non-additive interactions. B being some epistasis, C being a lot of epistasis. Now what you can see here is when we estimate the variance components, phenotypic variance increases a lot with the addition of epistasis. So an increase in phenotypic variance can lower the heritability of these traits. And we see that here in our heritability estimates, showing a pretty dramatic decrease in heritability with the presence of a lot of epistatic interactions. This can actually be expanded if you consider that variance, epistatic genetic variance can reduce standing additive genetic variance in the system. So we can see really striking reductions in heritability due to the presence of Y-linked epistasis. So what that really suggests to us is that Y-chromosomes having epistatic variance of this magnitude could really constrain the response to selection for traits. Immune traits for this, but the Y-chromosome influences a lot of things. And so that's what we're looking at now. Can Y or V reduce evolutionary rates due to experimental evolution? So we're excited to see those results come through. And with that, I thank people and thank you.